Thank you very much for inviting me and for hosting this event. This is clearly already the biggest and greatest Combine we've ever had. So this is really great to see. As Martin mentioned, I'm at Caltech, and I will tell you about recollections of how Combine got started and some of the goals from Combine, and maybe take a little bit of an assessment of where we are now. So a little bit about before Combine got started. So around the 1980s and 90s, personal computers, those of you who are old enough to remember, the personal computer became something that was affordable enough and powerful enough and common enough that many people became interested in using computers for various applications. And in the 90s, there was a lot of innovation in software tools for biological applications, and a lot of software development took place. People developed individual tools. Many of you in this room are authors of those original software tools. And so those tools initially were sort of islands onto themselves. They would read and write data files, but they were specific to those software tools. And it wasn't until, I would say, around in the 90s when the web took off and more software was being developed and people were attempting to share their results in more effective ways that they realized that maybe they should try to come up with common formats for exchanging models. And in the late 1990s, around 98 and 2000, a couple of these efforts made concerted attempts to develop software-independent formats for exchanging models in computational biology, and a certain set of domains in computational biology that we started calling systems biology, but also in physiology and sort of the neighborhood of that. And that was SBML, Systems Biology Markup Language, and CellML, one based in California, the other in Auckland, New Zealand. And initially we didn't know about each other, and so this was a sort of independent co-evolution, if you will. We in California were lucky enough that a project started by Hiroki Kitano was funded very generously by a Japanese agency called GST, Japan Science and Technology. And Hiroki Kitano got together with John Doyle, who's a professor at Caltech, and they started this project to try to develop one of the facets of this. So let me back up. Kitano's project was this overarching thing called Erato-Kitano Symbiotic Systems Project, and one of the things he did was infrastructure work for systems biology, and that effort was based in Caltech and Tokyo. In the Caltech group, we got myself, Herbert Sorrow, who's in the audience, Andrew Finney, were hired to develop software infrastructure to allow these many different software tools that had been developed in the 90s to communicate with each other more directly, so that people could use more than one tool in their workflow more effectively when doing data simulations, analysis, visualization, and that was a novelty in that time. So one of the first things we did was to basically try to gather people together who developed these software tools in the community and talk about how to achieve better interoperability. So this started a series of workshops with the catchy title, Workshop on Software Platforms for Systems Biology. And we did this starting in 2000, and for a number of years hosted about 2 workshops on this around the world. I unfortunately don't have pictures of the first couple of years of this. I may have them in film format, but this was also right around the time when digital cameras became affordable enough, and so I actually didn't have one until around 2002, 2003. But luckily, the University of Hertfordshire group, where our project lead, Hamid Boulouri, was located, and a number of other people, some of you here, were also at the university, working through the University of Hertfordshire. Two of the people there at the first workshop had a camera, and so I had photos from 2002, and I apologize for the quality. These are so low resolution, you would laugh. And I understand, some of you are probably wondering, like, didn't you have a cell phone or something? There were no cell phones with cameras back then, okay, let alone like an iPhone with a high resolution camera. So this is the best we could do. But anyway, these are really low res, whoops, sorry, pressed the wrong button. Low-res photos, and let me just point out a few people that are in this audience now. So Sven Saleh here, that's Akira Funahashi, looking very young and less gray. That's people from the E-Cell group, Kuichi Takahashi, and I'm blanking out on his name. I know his name, and I can't remember right now. Martin Gickel from University of the Max Planck Institute at Magdeburg. They worked on a system called Promotiva. This is the back of Ben Bornstein's head. He developed the first library for SBML. Over here, this is Hamid Boulouri. He sort of had a dual appointment between the University of Hertfordshire and Caltech, and he led our group on SBML development. This is Herbert Sorrow, who's in the audience now. That's Stefan Hoops, and yeah, I didn't check all of the registrations. I'm not sure if he's here, but anyway, he's with the Kopazi group, and still is involved with that. Over here, we have Autumn Quellar, who is from New Zealand, the SBML group. David Fell, who wrote this book on control of metabolism that many people probably are familiar with, and his, I can't remember, it was either graduate student or postdoc at the time, Mark Poolman. Yeah, no, that's what I said. I said Mark Poolman. I couldn't remember whether he was postdoc or a grad student. Andrew Finney, the mustachioed man here in low resolution. This is John Wagner from the Virtual Cell Group in Connecticut. And other people here, I'm not sure if they're in the audience, Nick Judy from EMBL EBI, and some folks I can't name, but many people I remember. So we kept doing these workshops. So this is one of them in 2002. We had another one later in the year in Sweden and Stockholm. I'm not sure of the wisdom of having a meeting in Stockholm in December, but be that as it may, we had, keep pressing the wrong button. We had this one in Stockholm, and that's Hiroki Kitano talking about, oh, this got really washed out, but visual notation for diagrams of cellular phenomena. That's Paul Nielsen from the SBML group. That's Hamid again. Joanne Matthews, she worked on early SBML website, the SBML web resources early on, back in those days. So anyway, a number of these workshops where people got together and talked about how to make interoperability work better between software tools and how to define standards that would allow that to happen. After about 3 years, there was a bit of shakeup of the Caltech team as people went on and got real jobs, and I didn't have anything else to do, so I just stayed and did SBML. And also, because the dominant conversation around the meetings started becoming SBML, we sort of hijacked it, and it became the SBML forum. And then later, we also introduced a hackathon series. So this ran from 2002 to 2009. The first SBML hackathon was organized by the Kopazi group at Virginia Tech in Blacksburg, Virginia. Stefan Hoops over here, that's me. Akira again. And this is the front of Ben Bornstein, who developed the first version of libSBML. And some other folks that you may recognize. And then later on, this is going forward, one of many meetings we had in Heidelberg here, actually. This was at the NH Heidelberg Hotel around the time of ICSB in 2004. And more people that, may or may not be in the audience at the moment. This is Claudine Chayowiya, who, with Denis Tsifri, helped develop the SBML, actually led the development of the SBML qualitative models package for exchanging models like PetriNet models, Boolean network models. And this gentleman standing up is Jim Schaff, who is one of the lead developers of Virtual Cell at the University of Connecticut. One of the reasons I wanted this particular photo here is because I don't know if you can tell, it's a little bit fuzzy, but I mean, you can sort of see the confused, confounded, possibly shocked expressions on people's faces. And I mean, there was a lot of that in those meetings. So we had these, we just had this all over the world. The hackathons, we alternated between hackathons and the forum meetings. One was for discussing standardization and applications of software tools. And then the hackathons were where people, it was more like CodeFest. People sat down, had software that they tried to get to communicate during the meetings and resolve software issues. And so by now, a lot of these faces are probably familiar because they were at many of these meetings originally. Here's Sarah Keating, who took over libSBML and has been the main developer for that since then. This is Robert Fair and his future wife, Anne Chasson. Robert told me that after this meeting, he proposed marriage to her. This was instrumental in this meeting. No kidding. He actually said that this was instrumental in him proposing marriage and them getting married. So SBML has been responsible for a lot of things. So as time went on, people realized they needed more than just the ways of representing the models. They needed also things around that. So annotations, standards, ways of annotating the representations to make them more precise, as well as, so that's, oh God, sorry, I keep pressing these tiny little buttons. This is a bad user interface, okay? SBO is systems biology ontology. Miriam is minimum information requested in the annotation of models, which is a minimum information standard for how to annotate models so that other people can understand what the models are about. And things grew, so NeuroML was another representation standard for tailored for computational models of neuroscience effects. And then SBGN for systems biology graphical notation for diagrams of network models in biology. And BioPax is a exchange format for pathway databases. A lot of pathway databases had no common way of providing that information. BioPax in the early years grew up around the same time and tried to standardize how pathway databases can provide their data. So many activities, and of course, these different standardization efforts also all had to have their own meetings. So we had more workshops. So now we started having workshops for the individual standards. So here's the first SBGN meeting for the graphical notation held in Tokyo. And many workshops and all, and time passed, and more standards. People realized they needed more standards, more standardization at the different for the different stages of work. So things to do with generation of models, describing the models, representing them, annotating them, how to describe what you do with the model, the analysis procedures, and how to present the results, how to represent the results in a way that is machine interpretable or human interpretable. Things like SBML are really important development because what they make possible is software independent ways of describing what do you do with the model to produce a particular result. And that makes it so that computational experiments can be more reproducible and machine interpretable. So there was a lot of this, there was sort of this explosion of activities in around the middle 2000s, and we also, because these things are not islands unto themselves, they had to be interaction. People had to discuss how do these things interact with each other, how do they overlap, and try to coordinate things a little bit better. So, of course, this led to combined workshops now. So we had super hackathons where many of the efforts got together for extended periods of time and talked about, well, if you have this standard, you have something in this standard, you want to communicate with this, and just work out more of the interoperability issues when you have multiple standards involved. And then we also had a combined CellML, SBGN, SBO, BIPAX, MyASI workshop organized by the CellML group, in particular Catherine Lloyd, the woman in the middle there. And this was held in New Zealand. And so these were actually really the precursors to what followed, because one of the things that became evident is we were having a lot of meetings, okay? I mean, just a lot of meetings. SBML alone, and since I was involved with SBML, I can only talk about the SBML things accurately, but I can tell you that in a 10-year period, we had 24 meetings, which is a lot of meetings to be having internationally. It's time-consuming for people because you waste a lot of time, during the travel. It's expensive. It's hard on people's schedules and personal lives, and it's bad environmentally because airline travel is actually a very bad thing to do in terms of carbon release into the atmosphere. So our carbon footprint was just really horrible for all these meetings. And also, it's just really inefficient, right? So we started having this idea into our heads that, couldn't we do a little bit better? And also, we started seeing some commonalities. Things would get repeated, like people who wanted to develop a new standard, a new way of doing something, representing something, would often just reinvent the same processes or similar processes from scratch, because there was nothing sort of off-the-shelf standard operating procedures or best practices available for them to reuse. And why am I looking at the screen when I have it in front of me? And then we had, there was also insufficient coordination between these efforts. Often limited per project funding meant that there was no continuity even between meetings, let alone providing support for infrastructure or additional activities related to standardization in this area, and no common identity or voice. Really, if you wanted to know about standards in the area, there was no single place you could go to find out more about them. And so we thought that a number of us thought that this was a bad situation. We started scratching our heads, and we thought, well, couldn't we combine our efforts to achieve greater harmony between... You're laughing at my bad jokes, aren't you? I know you're laughing at this, but it was true. It was genuinely true. This is really what was going through our heads. Like, can't we do better than this? So that's how Combine got born. And so Combine is Computational Modeling in Biology Network, and the goal really was to organize, to do a bunch of things. So organize community meetings, help coordinate standards development, develop common practices so people didn't have to reinvent things from scratch and can also learn from experiences of previous efforts, provide common infrastructure, excuse me, common infrastructure that could be, sorry. Thank you, good thinking. Common infrastructure that people could reuse for hosting standards, descriptions, and other information, and then basically act as a community representative for standardization efforts in this area. So the first Combine meeting was held at University of Edinburgh in Scotland in 2010, and the first Harmony, which is a hackathon on resources for modeling in biology, was held in New York City. It was organized by Emek Demir and Nadia Anwar of the Chris Sanders group at the Memorial Sloan Kettering Cancer Center in New York. So 10 years later, we're here, and thank you for being here and for continuing and being involved, many of you, for so long. We now have, as Martin mentioned, there are 8 standards that have they have well-defined open specifications. They have community-elected boards of editors, sometimes also scientific boards, and there are software tools to read and write these formats, and basically they have a certain level of maturity that you hope to see in a standard. There are associated efforts that are sort of supporting of standardization, the things in the first row, minimum information standards like Miriam, as well as standards for how to combine, put together files into single packages so that you can exchange it more effectively, permanent identifiers for things, and a bunch of related standardization efforts that are at various stages of maturity and development for different areas that include pharmacological, pharmacokinetics, pharmacodynamics models, things to do with neuroscience, Field ML, which is a long time effort from New Zealand, and a bunch of other of these efforts that have been going on for a while. How did we do? How do I think that we have done so far in the 10 years of Combine? So things have been a little slow. I think we've made progress in some areas. This is my take. Somebody else might rate these a little bit differently. But basically, in those 5 things that I mentioned as being goals of Combine, we've, I think, done quite well in organizing meetings that have reduced the number of individual meetings by glomming more of them together and try to get people to interact just by bringing them together. That's sort of the first step of getting people to interact. Oops. Then we have various efforts to have various ways to try to help coordinate development, but it could be better. I think we've achieved sort of like half goals on that. Develop common practices. We don't really have yet very many things that you can take off the shelf. There's a code of conduct that we recently started developing and have put out. There's a few other additional bits of information you can find on combine.org. But by and large, we don't have reusable procedures. What people have been doing is basically looking at what some other standard did and more or less copying that approach. Common infrastructure, I think we have reasonably well for that. And then as a community representative, we're sort of halfway there, but I think Martin has been really working at improving that situation, and you might have some things to say at some point, or certainly we'll be having discussions for more, let's say, official standing of Combined, a standardization organization. Can we do 10 years more? I think that's up to you folks here. So thank you. Thanks for being here. Thanks for listening.